Hi, I'm Hongkai Ji. I'm a professor in the Department of Biostatistics. I've been here since 2007, and so it's been over 18 years now. I first came to this department when I was on the academic job market, and I visited the department for an on-site interview, and I immediately felt that this is a very special place, and the people here are all very friendly, supportive, and truly interested in each other's work. So when I got the offer, it was a pretty easy decision for me. For me, I think it's the opportunity to combine statistical thinking with creativity to make a real impact on human health. And that combination is what I find most exciting. So I develop statistical, computational, and machine learning methods to analyze large-scale genomic data and use them to understand biology and human disease. My work sits at the intersection of statistics, machine learning, and new technologies, things like single-cell and spatial omics, and transcriptomics, epigenomics, and immune profiling. And at a high level, I integrate statistics and technology to answer a few foundational questions. One is how tens of thousands of genes coordinate across time and space to support normal development and function. And another one is how we can read signals from the immune system to understand a person's exposure history, like exposure to different virus infections and to thousands of bacteria and also different toxins, different allergens, and we are trying to reconstruct a person's exposure history using some accessible samples like blood samples, and then use that information to help us understand early life origins of human disease. And another area is understanding how our immune system responds to cancer immunotherapy and how we can use that information to improve patient outcome. And for all these questions, obviously we need to deal with new data, with non-trivial data structures. That's why we spend a lot of time developing new methods, such as methods that represent high-dimensional genomic data in a low-dimensional space using non-traditional structures like spanning trees to help us infer cells' developmental trajectory, and such as methods that use Markov models to infer cell state transition dynamics in our immune system, and also such like methods that help us to train better neural network and large language models using sparse and noisy single cell and spatial genomic data. Yeah, I hope our work produce methods and tools that many people can use to better understand genomic and cellular data. And more broadly, I'd love for some of our findings to contribute to foundational knowledge in life sciences and ultimately help improve disease prevention and treatment. That's a good question. I think we are moving towards a future where we can measure a biological system more comprehensively and more dynamically, and such as creating a movie of cells over time with information collected at multiple levels from DNA to RNA to proteins. And at the same time, AI models trained on massive amounts of data will become much more powerful in predicting cells' behavior and how they will respond to perturbations. And this will allow us to build digital twins of biological systems like virtual cells, and which will be incredibly useful for both basic and translational research like drug development. I would say the people is a community that is kind of passionate about science, open-minded, and very supportive. So I recently stepped down as graduate program director, and I was in that role for about 8 years, from 2017 to 2025. And it was a great experience, especially being involved in shaping the PhD program and recruiting students. And I stepped down last summer, and now I'm focusing more on teaching and mentoring. And I teach PhD level courses on advanced methods, and also I spend a lot of time working with my students and postdoc on their research and their career development.